Enzymes. Enzymes are proteins that speed up a chemical reaction. The induced fit model of an enzyme action refers to the interaction between the enzyme and the substrate. The substrate binds to active site of the enzyme due to its specific shape. Here shows an induced fit model. When a substrate molecule collides with an enzyme, if its shape is specifically correct, the shape of the enzyme's active site will change so that the substrate fits into it and an enzyme-substrate complex can then form. The properties of an enzyme in a tertiary structure of its active site. All enzymes are globular proteins with a specific shape. They are usually specific to only one reaction. This links to the lock and key theory. The overall 3D shape of an entire protein molecule is a tertiary structure. The protein molecule will bend and twist in such a way as to achieve maximum stability or lowest energy state. Although the 3D shape of a protein may seem irregular and random, it is fashioned by many stabilizing forces due to bonding interactions between the side chain groups of the amino acids. Lock and key hypothesis is a model how enzymes and the substrate join. It states that the shape of the active site of enzymes exactly combine to the shape of the substrate. When a substrate molecule collides with an enzyme whose active site shape is combined, the substrate will fit into the active site and an enzyme substrate will form. The enzyme will catalyze or also cause a reaction, and the products, together with the enzyme, will form an enzyme-product complex. According to this model, it is possible for an enzyme to catalyze a reverse reaction. Here shows a diagram of the lock and key theory. Factors that affect the rate of enzyme reactions. Temperature. Enzymes, as you know, are proteins that speed up chemical reactions in our cells in our body. Enzymes work best at the optimum temperature, which means the best temperature. This is why homeostasis, balance of everything in a body, e.g. water, is important to keep our body temperature at a constant 36 to 37 degrees. So, as the temperature increases, so does the rate of chemical reaction. This is because heat energy causes more collisions with more energy between the enzyme molecules and other molecules. However, if the temperature gets too high, over 40 degrees, the enzyme is denatured and stops working as the active site is damaged and cannot work/function anymore. The pH scale. As you know, different enzymes work at different optimum pH values. This is a pH scale: acid, alkaline, or neutral. As you increase the pH scale, it can be denatured if the original pH is changed to a different pH instantly or gradually. Changing concentration of a substrate only can affect the rate of reaction if it's a limiting factor. That is, the factor that is stopping a reaction from proceeding at a higher rate. As the process goes on, then it will mean that substrate will get used up. Increasing substrate concentration increases the rate of reaction. This is because more substrate molecules will be colliding with enzyme molecules, so more of the product will be formed. However, after a certain concentration, any increase will have no effect on the rate of reaction, since the substrate concentration will no longer be the limiting factor. Increasing enzyme concentration will increase the rate of reaction, as more enzymes will be colliding with substrate molecules. However, this too will only have an effect up to a certain concentration, where the enzyme concentration is no longer the limiting factor.